Welcome biologists to part 2 of chapter 7a. So we're going to shift our focus now to chromosomes. So we're going to learn about chromosomes in this video, which will be shorter than the first one. You'll know the role and structure of chromosomes and how they're paired, and you should be able to describe chromosomal abnormalities shown by karyotypes. So in our study design, these are the key areas that we're looking at: the nature and pair of a pair of homologous chromosomes, the variability of chromosomes in terms of size and number, and karyotypes and what they can tell us is really what we're going to look at. So in this video, you'll understand the definition, structure, and function of chromosomes. You'll be able to define a homologous chromosome, differentiate and define autosomes versus sex chromosomes, define karyotypes and what they can tell us, and define the key chromosomal disorders that you need to know. So, genes to DNA to chromosomes. We know about genes and DNA. We looked at that in the previous video. But what we also need to understand is that DNA, which is a big molecule, needs to be stored inside tiny cells, and not only just tiny cells, but within their nucleus. So one strand of DNA, if you unwound it, is about 2 meters long, but it needs to be packaged so that it fits inside the nucleus. So how does that happen? So DNA in a diploid cell is about 2 meters long. The molecule, though, is very, very skinny, only 30 micrometers or 30 millionths of a meter in diameter. So how is it packaged into a chromosome? It supercoils around histones. These histones supercoil on top of themselves to form a nucleosome. The nucleosomes then coil again to form chromosomes. So basically coils are coiled around coils, which are coiled around coils, and that leads to a chromosome. It's kind of how it works. Okay, so if we start here, these are both showing the same thing. So in a broad sense, here's your long molecule of DNA. The DNA is wrapped around these proteins called histones. The combination of DNA and histones forms something called a nucleosome. So it's coiled around the histones. The histones coil on top of themselves, which is what you can see here, and they coil ever tighter until ultimately they create the chromosome, tightly packaged DNA, which is then put or stored within the nucleus of the cell. So this is just a blown up image of that sort of first couple of steps, or it's a simplified diagram even. You've got your DNA coiled around the histones. Those histone DNA complexes are called nucleosomes, and then they coil on top of themselves to form the chromosome. Very similar in prokaryotes, so your bacteria. So we've already talked about this in eukaryotes, which is like us, histones, and then chromosomes. But a prokaryotic cell just typically has one circular chromosome, but it's just packaged DNA tightly into that circular chromosome. Okay, so we know what a chromosome is. It is the packaging for DNA. We understand how it is structured and how it is created. So now let's have a look at the structure of each chromosome. In our body cells, or somatic cells, we have 46 chromosomes. Each one of the chromosomes varies in size and structure. Each chromosome will carry genes that are specific to that chromosome number. So we have 22 pairs of autosomes, or non-sex chromosomes, and 1 pair of sex chromosomes. Those 22 autosomes are numbered 1 through 22. And each one of those has its own unique set of genes. So chromosome 1 has a set of genes. Chromosome 2 has a different set of genes. The overall structure, you've got the telomeres, which are it's on the end of these chromosomes. This one here on the left is one chromosome. It's one copy of the chromosome. And on the right, here's the second copy of the chromosome. So this is actually 2 chromosomes, and they're joined together at what is called the centromere. Because they are the same, so let's say this is chromosome number 1. These are referred to as sister chromatids. So they're copies of the same chromosome. Joined together at a centromere, you've got DNA within it, which we already know, and there's a short arm and a long arm, which I would think is pretty self-explanatory. So homologous chromosomes, that's 2 copies of the same chromosome. 1 copy from mum and 1 copy from dad. They're the same size, they're the same structure, and they have the same genes located at the same points on the chromosome. So say you have a gene for blood group that's right here at the very tip on the telomere on chromosome number 1. It's going to be found at the tip on this chromosome copy and the tip on this chromosome copy. Not only that, but if this was my chromosome number 1, if we looked at your chromosome number 1, you'd have the gene found at exactly the same location as well. Might be a different allele, but the gene will be found in the same location. And it's always found in the same location on what is referred to as homologous chromosomes. That is, chromosome 1, the copy from mum, the copy from dad, they're homologous. Chromosome 5, the copy from mum, the copy from dad, they're homologous. Make sure that you understand that. Homologous, or homo, meaning same. Same chromosome, basically, is what we mean, yeah? Hopefully that makes sense. So homologous chromosomes, we've already said, they're the same size and length. They have the same centromere position, so in other words, where they join, and they share the same genes at the same loci. And so what we're seeing here, chromosome from parent 1, mum, parent 2, dad. You might have different alleles, blonde hair versus brown hair, but the gene is found at the same location. Hopefully that's fairly self-explanatory and makes sense for you. All right, so then we can actually look at our chromosomes and what chromosomes we have within each cell by doing something called a karyotype. It is a visualization of all of the various chromosomes within a particular cell. So you can check the size of the chromosomes, the structure of the chromosomes. You can also look for banding patterns on the chromosomes. They are arranged by size. So chromosome number 1 is the biggest, down through to chromosome 22, which is the smallest of your autosomes. And then you've got your sex chromosomes, X and Y. So they're in the red circle. For a male on the left, you have the X and the Y. They are not homologous because they're different chromosomes with different genes. But for a female, the X and the X, same chromosome, same genes, homologous. Autosomes versus sex chromosomes, 23 pairs of our chromosomes. 22 of those are not related to sex, in other words, whether you're male or female, and they are referred to as autosomes. So an autosome is a non-sex chromosome, and they are pairs numbered 1 through 22, and each of those are homologous. Sex chromosomes homologous, we've already said they can be. All right, so in a female here, they are, but if this was a male, they're not homologous because you'll have an X and a Y. So your sex chromosomes are your X and Y chromosomes only. What can karyotypes tell us? Well, they can tell us the gender. So here, there's 2 X's. This is a female individual. They can also tell us about chromosomal disorders. For example, here, there are 3 copies of chromosome 21. We're only meant to have 2. This is a condition referred to as trisomy 21 and refers or relates to Down syndrome. And so we're going to have a look at those in just a moment, the various syndromes that you can tell by looking at someone's karyotype. So karyotypes can tell us the different species because different species have different numbers of chromosomes and their chromosomes are structured differently. They can tell us about aneuploidy, which is where you gain or lose a single chromosome, like trisomy 21. All right, so you've gained an extra copy of chromosome 21. That is referred to as aneuploidy because you've gained an extra one. Or if you lose a chromosome, so they only had 1 copy of chromosome 21, that would also be referred to as aneuploidy. Polyploidy is where you get a completely an extra copy of all of the chromosomes. All right, so we're going to have a look at tetraploidy in humans. Tetra means 4, so it means you've actually got 4 copies of the chromosomes instead of the usual 2. So looking at some of the syndromes, Turner syndrome, phenotypically they are female. In other words, the phenotype is the visible trait, so they look like they're a female. But their genotype, in other words, what chromosome will make up they have an X chromosome and then nothing. All right, so this would be an example of aneuploidy, where they've lost a chromosome. So they've only got 1 sex chromosome and it's an X. There's a number of features that you can see in this diagram, but some of the key ones, they are short. Typically, they are only about 4'8". They have cognitive issues, a webbed neck. They are infertile, often have cardiac problems, and often have moles all over their body or brown spots. Klinefelter syndrome, again, phenotypically, they're male, but this time they've got an extra chromosome, 2 X's and 1 Y. Some of the traits that you'll see, they are taller than average. Again, they can have cognitive issues, incomplete puberty with enlarged breast tissue, less body hair than usual, reduced muscle mass, and weak bones. Down syndrome we've touched on. It's caused by an additional copy of chromosome 21. So again, aneuploidy. In this case, an extra copy of chromosome 21 or trisomy 21. Again, a number of features that are outlined here, but some of the key features: a broad flattened face, almond-shaped eyes, short neck with small ears, tongue that typically protrudes, weaker muscles, and again, cognitive impairment. And tetraploidy. So this is a karyotype of someone with tetraploidy. 4 copies of chromosome number 1, 4 copies of chromosome 22, etc., etc., etc. So it's a rare abnormality where each cell has 4 copies of each chromosome instead of 2. Usually that ends in miscarriage. It's not compatible with life. However, sometimes they do survive to be a live birth. And when this occurs, there's a number of abnormalities, which includes growth retardation and failure to thrive, eye issues, heart issues, kidney issues, and genital malformations, and also limb defects. Just to go back on one other thing I said about karyotypes, which is that you can actually determine the species, often based on the karyotype. Different species have a different number of chromosomes. So our closest living relative, chimpanzees, have 48 chromosomes. We have 46. So by looking at the karyotype, it can help you differentiate. And that's all this slide is really aiming to tell you. So again, a reminder of the key features of our study design, which this video has aimed to tick off. And hopefully you can now explain the role and structure of chromosomes and how they're paired, so particularly homologous pairs. And you should be able to describe chromosomal abnormalities as shown by a karyotype. If you have any questions, as always, do let me know. And I do hope this video was helpful.